in this video I will show you how to do the conjugation. So this is part of the transformation and conjugation lab, and this is the first session of conjugation. So conjugation is a form of horizontal gene transfer. Here the DNA is transferred between bacteria cells by cell-to-cell contact. So we are going to transfer the plasmids that was first introduced into the E. coli cultures into our Flavorbacterium johnsonii mutant. So we have our mutant culture and we have our two E. coli cultures, each containing a different plasmid. So we have PDH223 and PSA11. So these two E. coli cultures, they have these two plasmids. So I have measured out 1 milliliter from each of these three cultures. So now we're going to do, what we're going to do is we're going to mix these so that they have, they will contact with each other, and then there will be plasmid transfer. So before doing that, there are several steps. So first we have 1 milliliter of the cultures, and now we're going to spin it down and separate the cells from the culture. So I'm going to spin this inside each. So you have this centrifuge, you have seen how the centrifuges work before. So we need to remove all the media and separate out the cells. So we will spin this. So if you remember, we need to balance when we put the tubes in the centrifuge, the weight should be balanced. And now I'm going to close this and we will spin this for 1 minute at 10,000 RPM. So after centrifuging step, we have a supernatant and we have the cell pellet. So now we need to remove the supernatant and we need the cell pellet. So I'm going to remove the supernatant, or the liquid part. And when we do this, we need to be very careful not to break the cell pellet. After removing the supernatant, we will do a washing step for the E. coli. The reason we do this is because if you remember the two plasmids, they have ampicillin resistance gene. So when we grow these two E. coli cultures, the media contains ampicillin. So before we mix our E. coli with the Flavorbacterium johnsonii mutant, we need to remove all the ampicillin. Otherwise, the Flavorbacterium, who doesn't have ampicillin resistance, will be killed by any residual ampicillin. So to wash wash these two cell pellets, we're going to use fresh, clean CYE media. CYE media is has the necessary nutritional requirements for Flavorbacterium johnsonii to grow, and E. coli can also grow in it. So that's why we use CYE. So this is fresh, sterile CYE. So I'm going to use 1 milliliter of CYE and resuspend the E. coli cell pellets. So when we resuspend, we need to be very gentle. These E. coli cells have special villi that they need to attach to the other cell when they transfer the DNA during conjugation. So if we do like a fast pipetting or vortexing, these villi can break. So we don't do any vortexing, and the resuspension, we only do gentle pipetting. So now these pellets are resuspended in clean CYE media, and now we can centrifuge these again to get a cell pellet. And for the Flavorbacterium johnsonii, there are no antibiotics added into the media, so we don't need to do a washing step for them. So they will be resuspended in 60 microliters of CYE. So before we spin the E. coli down, I'm gonna resuspend our Flurobacterium cell pellet in 60 microliters of CYE. So this is set to 60. I'm gonna take 60 microliters of CYE and resuspend our Flurobacterium pellet. Right, now we only spin the 2 E. coli cultures, not the Flurobacterium. We're going to spin again in the centrifuge, the same time and the same speed. So 1 minute in 10,000 RPM. Alright, so after the centrifuge step, we have the 2 E. coli cultures. I have removed the supernatant and I resuspended both of them in 60 microliters of CYE. Now we have all 3 cultures resuspended in 60 microliters of CYE. Now we're going to do different combinations. So I have labeled 4 tubes, one as Flurobacterium johnsoniae, FJ, and one EC for E. coli, and one as FJ, PSA11. So here in this tube we will mix the Flurobacterium with the PSA11 containing E. coli, and this is labeled as FJ PDH223. In this we will mix the FJ with the E. coli containing PDH223 plasmid. Now I'm going to measure 20 microliters of FJ into this fresh tube. 20 microliters. So this is our first negative control. Change the tip and measure 20 microliters from the E. coli PSA 11. And 20 microliters of E. coli with PDH 223. So this also goes in the same tube, it doesn't matter. This is our E. coli control. And now we're going to mix the FJ with the 2 E. coli. So measure 20 microliters of FJ. So I'm going to measure into both tubes, because then I can use the same pipette tip. Okay. Now I'm going to mix 20 microliters of E. coli with PSA 11 in the tube labeled as PSA 11, and another 20 microliters into the other tube. So now we have 2 controls and 2 of the conjugation mixtures. Now we're ready to plate them. So I have labeled 4 CYE agar plates according to what we have written on the tubes. So these are corresponding to what we have mixed in these 4 tubes. So there's FJ in CYE. This is for our negative control, and this is EC for another negative control, and these 2 are our conjugation plates. Now I'm going to transfer the mixtures in these 4 tubes onto our agar plates. So here we do what we call spotting. So we're not going to be spreading or streaking these liquids on our agar plates. We're just going to spot them. So I will show you how we do it. So I'm going to take the whole amount that's in here, in this FJ labeled tube. There's 20 microliters of FJ, so I'm going to take the whole amount and I'm going to place this as a drop in the center. So I'll explain why we do this. So in our EC tube we have 40 microliters, so I'm going to change this to 40 and then transfer the whole 40 microliters onto this agar plate. So having the liquid, the mixture as a droplet is important for the conjugation, because we need to have all our cells in close proximity so they have maximum contact. So conjugation happens as a result of cell-to-cell contact, so we need to provide enough, provide the space and actually less space, so then they have more chance of contacting each other. So that's why we don't spread or streak on the plate. Instead we place it as a droplet or a spot. So after doing this, after spotting our plates, we are going to let them dry for a short while, and then we are going to put these in the 30-degree incubator for 2 days.